Assalamualaikum and a very good day. This is a video for experiment 4.1 digestion of protein. Digestion is the mechanical and chemical breakdown of food into small organic fragments. It is important to break down macromolecules into smaller fragments that are of suitable size for absorption across the digestive epithelium. A large part of protein digestion takes place in the stomach. The enzyme pepsin play an important role in the digestion of proteins in stomach, which have acidic environment by breaking down the intake protein to peptides. In small intestine, specifically duodenum, which have an alkaline environment, trypsin acts on peptides, reducing them to smaller peptides. The requirements for this experiment are 6 test tubes. All test tubes will be labeled as P2 for pepsin at pH 2, P9 for pepsin at pH 9, and PC for control for pepsin. T2 for trypsin at pH 2, T9 for trypsin at pH 9, and TC is control for trypsin. pH paper, pipette, 1% pepsin and 1% trypsin with measuring cylinder and dropper, 0.1 molar hydrochloric acid and 0.1 molar sodium carbonate. Egg white and water bath that has been set at 37 degrees Celsius. Measure 5 ml of pepsin using measuring cylinder and dropper and put it into P2 test tube. Repeat the procedure for P9 and PC using pepsin. Using different dropper and measuring cylinder, repeat the step using trypsin for test tube T2, T9, and TC. Then, pipette 0.5 ml HCl and put it into P2 test tube. Repeat the procedure for T2 test tube. Then, pipette 0.5 ml sodium carbonate and put it into P9 and T9 test tube. To check the pH of the content for all test tube, dip the pH paper into the solution and compare the color change at pH paper with the color chart to make sure the solution at the correct pH. Next, put small strip of egg white into all test tube. Next, put all of the test tube inside water bath carefully using tongs and leave for 1 to 2 hours. After 1 to 2 hours, take out all the test tube from water bath and observe the appearance of egg white and the surrounding solution. Then, record and interpret the observation in table 4.1 in the lab report.